So we will begin with Amanda MacFarlane. I don't know where she's sitting this morning, from Health Canada, who'll be talking about the type and strength of evidence needed for determining special nutrition requirements. Thank you, May.
Thanks for having me today. Thanks to the Planning Committee for inviting me to speak on this topic. I'm going to reflect a little bit on the dietary reference intakes and what we can learn from the dietary reference intakes when we're thinking about what kinds of evidence you could potentially use when looking at special nutrient requirements in disease states. Now, I do this unapologetically despite being Canadian. Someone at the microphone yesterday mentioned about apologizing for talking about the DRIs, but I think we don't need to apologize about the DRIs. There's lots to be had. So these are my conflicts of interest. So I am a research scientist at Health Canada, which is a sponsoring agency, and I'm a member of the Canadian Federal Steering Committee for the DRIs, as well as the chair for the Joint Canada-US DRI Working Group. So looking at the DRIs, I want to go back to I'll reflect a lot on what Patsy Brannon started to talk about yesterday morning. And I think that was the perfect jumping off point for what I'm going to be talking about today. So we know that the DRI values are based on nutrient intakes and indicators, especially when we're talking about indicators of adequacy. So the whole point of the DRIs is so that we can, from a public health perspective, avoid nutritional deficiencies in our population. The DRIs also have some focus on adverse effects of high levels of intakes of specific nutrients as well. And everybody is familiar with the concept of the upper tolerable intake level. So the data is based on data from apparently healthy populations, and therefore they apply to apparently healthy populations, which differs obviously from what we've been talking about the last couple of days. The DRIs do take into consideration the concept of chronic disease risk reduction, and it has always been with the caveat of where sufficient data for efficacy and safety exists. And this is an important notion that only recently really we have some guidance on how you would actually apply those, and I think the current sodium potassium committee is working diligently on that. So the DRI approach has some fundamental aspects that could be adapted and potentially applied to special nutrient requirements in disease states. And I do think that the case studies, for instance, that were presented yesterday, it makes the case that there probably are some special nutrient requirements in disease states. But because you have, you know that a relationship exists, doesn't mean you can base standards on that. You need a certain level of evidence, a certain type of evidence, in order to make standards by which you would that you would then apply. So the first step in the risk assessment approach for setting DRIs is the demonstration of causality. So this is your hazard identification. In the absence of a causal relationship, you really can't move forward. And so this requires a literature review, and in this day and age, that means a systematic review and hopefully a meta-analysis of the literature. It means interpretation of the data. And of course, it also means identification and selection of the appropriate indicators or endpoints. And Patsy talked about them being health outcomes, and with the DRIs, I think that's probably a better description of the kinds of endpoints that we're thinking about. Once you have confidence that there is a causal relationship between the intake of a particular nutrient and a disease of deficiency, when we're talking about the DRIs, then you need to be able to model the intake-response relationship. So this data is different. You need data at multiple doses that then have an association with the endpoint of interest. Once you can establish that model, you can actually, as Patsy pointed out, mathematically derive your DRI values for that. Now, the DRIs didn't just leave off at setting the intake values. They also went about looking at the intake assessment of the two populations of concern, which for the DRIs are Canada and the US. And then there's always a chapter in the DRI books about implications and special concerns. So this is not a new concept, that there may be certain conditions or physiological states that require special consideration in terms of nutrient intake. So, can the DRI risk assessment framework apply to nutrient requirements in disease states? And I would argue that there are some fundamental aspects of that DRI risk assessment approach that are fundamental to establishing special nutrient requirement intake values in disease states. So first, let's talk about establishing causality and identification and selection of the indicators. This is a critical point. So when you're establishing adequacy, you have a very high level of confidence that there is an association between the exposure or intake of a given nutrient and the clinical outcome. So the example I have here is for folate, my favorite particular nutrient, where you have folate intake, you know that that modifies your indicator of status, RBC folate, which is directly linked to the clinical outcome, which is megaloblastic anemia. And it's the same for vitamin C and scurvy, vitamin D and rickets. And so you have a high level of certainty that the nutrient is having a causal relationship to whatever endpoint you're looking at. So when we talk about the concept of nutrient intake as it relates to chronic disease, it's still in the same direction. It's still in the same direction where you want to associate an exposure or intake with a clinical outcome, but it's way more complicated. And so basically what ends up happening is you have a lower level of certainty of that relationship between the exposure and the clinical outcome or chronic disease, mostly because you never are able to associate that clinical outcome. You often will rely on the use of validated surrogate outcomes, or sometimes in the literature you'll see lots of non-validated intermediate outcomes, and this just adds to the degree of uncertainty you have of the relationship between your exposure and your clinical outcome. So how do these relationships change when we're considering disease state specific nutrient requirements? And I would argue that the direction in its simplest form it's different. Instead of nutrient intake being associated with a disease of deficiency or a chronic disease outcome, you're now looking at the disease state is occurring, which can impact exposure and or the indicator of status and or the clinical outcome associated with that indicator of status. And the relationships are not always going to be straightforward. So the disease could have a direct impact on the outcome, or vice versa. And then, of course, the disease itself, as we saw yesterday, could actually alter the biomarkers of status. And so this relationship, all of a sudden, it's clear, it's much more complex than what we're used to dealing with when we're looking at nutrient intakes and diseases of deficiency. So I would thank the speakers from yesterday because you allowed me last night to go back and fill in some of my blanks on these proof of concept examples. So don't take this as being an end-all be-all. These are really proof of concepts here. So just giving an example of IBD. So we know that IBD results in intestinal inflammation and malabsorption. We know that it's associated with low B vitamin status or iron status and associated with anemia. So the question is, if you're going to set a special nutrient requirement, do you know that the exposure or intake is going to affect your status indicator, is going to affect your anemia? So what's your outcome? Because for the example of iron, your outcome could be the anemia, or it could be the inflammation or malabsorption. So we have to start making decisions on what your outcome is. And then, of course, the exposure might be different. When we talk about the DRIs, we're talking about dietary intake. But when we're talking about disease states, your exposure could be IV, it could be oral, it could be IP, and the iron form could make a difference here. So again, it just adds to the complexity. And every time you add complexity, you're increasing your level of uncertainty. Another example here, it's even more complicated, is the example from CF. So here you have your CF gene mutation. It's related to pancreatic insufficiency. It's related to low status for fatty acids, fat-soluble vitamins, proteins, and carbs. Those all can be related to growth and bone health, which would be traditionally, for the example here of vitamin D, a fat-soluble vitamin, that would be the traditional DRI health outcome associated with inadequate vitamin D. But here we heard yesterday during the talks that growth is associated with the CF outcomes, and we know that growth is also associated with lung function. So then it becomes the question again, what's your outcome of interest that you need to focus on? Is it going to be growth? Is it going to be your traditional bone health outcome? Or potentially, is it even going to be lung function? So you have to be transparent, and you need the data to back it up. So changing gears a little bit here, I'm going to talk about the data required for estimating intake-response relationships. So again, looking at when you were considering intakes associated with disease of deficiency, we think of it in terms of there's a threshold, and there's a threshold between what would be considered inadequate intakes, and a threshold above that threshold would be considered safe and adequate. So traditionally, the gold standard for setting these dose modeling and establishing causality, ideally you want that top level of evidence, the highest level of evidence, and that would be data from RCTs. And in an ideal world, it would be a systematic review and meta-analysis of those RCTs. The DRIs have relied heavily on supporting study types such as intervention trials or metabolic balance studies, depletion-repletion studies. But it should be also noted that in order to do dose-response modeling, you do need studies that have more than one dose. So you need at least 3 doses in order to do that modeling. And then once you have that data, that's how you're able to then estimate your requirements. And then, of course, the RDA is mathematically derived from the EAR, so it's dependent on that as well. So in theory, the approach of setting an EAR and an RDA could apply, I would say, arguably to highly prevalent diseases, mostly because those diseases will have more data, more evidence, and so might be more likely to have the kind of evidence you need to establish an EAR. But I think the case is probably true for a lot of diseases, is that either they're rare or we're lacking a lot of the evidence. So instead of having a population's worth of evidence, maybe we have evidence on a few populations. This population tends to be me when I used to be blonde. It changes on a regular basis. So all of a sudden, estimating an EAR becomes less feasible because you just don't have the data to support it. And so any approach to setting special nutrient requirements really needs to consider the likelihood that there may be a paucity of evidence. So what do you do in that case? And this is mostly questions, and I think any future committee has to grapple with these questions. And that is, so if you have lower level of evidence, is that acceptable? And if you have a lower level of evidence, that means you automatically have a higher level of uncertainty. And so that means you have higher level of uncertainty around the causal relationship and the ability to have a dose-response modeling. So I just bring in this topic. There might be acceptable novel study designs that could help bridge some of this lack of evidence. And hopefully I'm looking forward to the talk in a couple of talks about that. And then I want to talk about, one of the last things I want to talk about is the concepts of harm as it applies to the DRI risk assessment framework. So the DRIs, we don't only focus on the low end of intakes, which has the higher risk of disease of deficiency. We also have this upper tolerable intake level above which there's potential for adverse effects. In the DRI approach, there's also an assumption that you have an interval between the RDA and the UL, such that there's a safe interval of intakes. I think it could be argued that that might disappear when we're considering special nutrient requirements in disease states. And I've just given a couple of examples here of some diseases where nutrient intakes in the what would be considered the healthy range may actually pose a harm, and that's potassium in kidney disease, or protein in PKU, or supplemental iron in hemochromatosis. And then, of course, there's the potential for overlap. So I've stolen this figure from work that was done for developing the principles for using chronic disease endpoints and setting DRIs, where we're looking at multiple background risk for 3 different diseases, and as nutrient intake increases, you could have decreasing risk of one disease but increasing risk of another. And then, of course, there comes judgment. What do you do when you have these intersections? And so I could foresee that in the concepts and framework associated with special nutrient requirements in disease, you could have something very similar. And the example I give here is low protein diets for PKU, which can result in inadequate intakes potentially of other essential amino acids or minerals and vitamins. Dr. Falk gave a really good example yesterday in her talk where she said that people are proposing ketogenic diets for people with mitochondrial disease because it has beneficial effects on mitochondrial function, but then it could have dyslipidemic effects and exacerbate their existing dyslipidemia. So these are considerations. And then, of course, how do you define special or distinct, whatever word you're using? So is it simply a matter of a shift in the distribution? So here is your disease EAR lower than your healthy EAR. And if it is that simple, how different does it have to be distinct? And I think this is where the judgment comes in, and I think as we move forward, this is really what's going to have to be a focus. And the reality is that it may not only depend on differences in distribution of requirements. There's going to be a lot of other considerations that will have to take place. And so the first one that I've got here that might influence the distinctness of a nutritional requirement would be the prevalence and severity of disease. If you have a rare disease but has a very severe outcome, you may accept a lower level of evidence because of the consequences of not doing anything. But that might not always be the case. And then, of course, we talked a lot about disease activity. If you're in remission versus an active disease state, that might influence. And then, of course, inflammation. And then the effective dose needs to be known. The effective route needs to be known. The matrix that works best. Gender and life stage will influence these values. Nutrient form, whether it's synthetic or natural. Genetics of the individuals. And extrapolation. So in the DRIs, we have done a lot of extrapolation from one age group or life stage group to another out of necessity. But in the case of special nutrient requirements in disease, we need confidence that everyone's equally responsive if you're going to make these recommendations. And then, of course, there's the concept of maintaining nutritional balance. If you're taking away some nutrient, is that affecting adversely the nutritional balance of other nutrients? These are really important considerations when defining special nutrient requirements. So in summary, I would argue that aspects of the DRI risk assessment approach are certainly applicable and adaptable to determining special nutrient requirements in disease states. But I think any adaptation of the approach has to be transparent. And I think we have to be very careful about defining who's the clinical population that will be effectively treated with such nutrient requirements. What are the outcomes and endpoints that are going to be used? What's the acceptable level of evidence? And if there's a modifier of the acceptable level of evidence, what it is. We need clear and transparent analysis of the risks and benefits, especially when they overlap. We need rationales for extrapolation, if they're even acceptable. And then we also need the inclusion and definition of any special considerations when setting these kinds of nutrient values. And I would argue my last statement here is if you build it, it doesn't mean they'll come. So you can build a framework, but you can't presume that you have the right kind of evidence to actually make nutrient requirement values. So let's keep that in mind. And with that, I'd just like to acknowledge the working group members who have been keeping me invested in the DRIs for a number of years now, the Workshop Planning Committee, and Patrick Stover and Martha Field gave me some feedback on the presentation. Thank you.